Okay, student. My name is Mr. Laurent Ndonje, a biology teacher. Today, I'm going to continue presenting to you a topic of ecology. Our topic is ecology. That is our main topic. In the last lesson, we started a subtopic of methods of studying ecology. And today, we will continue looking on that part. Methods of studying ecology. My student, I'm sure you remember on the last lesson I presented to you the techniques used in sampling. I talked about random sampling, systematic sampling, and stratified sampling. I talked about when you have a population having organisms of different characters. I said we use stratified sampling, where the population is divided into several subgroups, then from each subgroup a sample is taken. I remember we talked about that. And also in the last part of that lesson, I said that in this lesson we will discuss the use of transect and quadrant in sampling flora and fauna. Therefore, today we will see methods of sampling, but specifically now the use of what, student? Transect and quadrants. The use of transect and quadrant. My student, let us start with the transect. Try to think, what is the transect? My student, for example, you go into the field, you have a string. Let's say you have a string. A string will join 2 points. For example, you have selected a point. Mind you, student, the point are to be selected by the researcher. You have selected a starting point. Let's say this is a starting point which you have selected. And this one here is the ending point. Therefore, you find a string is placed here. You see now? Therefore, this is a transect. We call a line transect. Why do we call transect? Because it can be used for a longer distance. It can be used for a longer distance. You must have 2 points. But sometimes, my student, you can have a certain area where you can put 2 lines transect. Therefore, you put 2 strings. For example, this is a starting point which you have selected. This is also the other point which you have selected. Here now, you see, my student, this is also another point. This is a line, this is a line. Therefore, here, the organisms which are here, are recorded. Therefore, are recorded. But this line are running parallel. There are 2 parallel running lines. Parallel running lines. Therefore, in a transect, a loop or a tape is used, but at a specified distance. Therefore, you must specify distance before starting to sample. My student, for example, you are sampling plants. Maybe, let's say, of the distance of 500 meters. You wanted to use now a line. Therefore, you must establish a starting point, then a regular interval that I will be sampling at this interval. Therefore, a transect is used when sampling systematically. Hope you are following, my students. Now, from these examples, what is a transect, my student? I see is a line. This is a line or a strip between 2 points along which ecological investigator conduct sampling. Hope you are following. Therefore, my student, from the explanation which I've given you, a transect now is a line or a strip. Now, but this a line or a strip is between 2 points. Between 2 points. Now, along which an ecological investigator conducted the so-called sampling. My students, a line between these 2 points. Therefore, we have a point here, we have a point here. A line along which an ecological investigator conducts sampling. Therefore, sampling will be done here on this line. Are you following, my students? A strip. Therefore, this one here, a line. Okay? Now, a strip, my student. A strip. You find now here, touching there. A strip now. You find 2 lines. But these 2 lines, they have a specific width. Maybe of 0.5 meter or of 1 meter apart. Are you following, my students? Hope you are following. Therefore, my student, when you want to use a transect, it's simple. Have your tape measure or have your string. Place it on the ground to the area which you want to sample, specifically plants. Then organisms touched by the line are recorded. You take them as a sample. Are you following, my students? Now, from the definition here, we get 2 types of transect. There are 2 types of transect. Therefore, type C of transect. There are 2 types of transect. One, my student, is called line transect. Line what? Transect. In line transect, a string or a tape is laid on the ground. Are you together, student? And then organisms touched by the line or a string are recorded. Now, take an example you have this point here, you have this point here. Therefore, as an ecological investigator, you will be counting the organisms which are touched by the line. But remember, you must have a specified distance. Specified distance. For example, maybe of a specified distance of 200 meters is the area which you are sampling. Now from there, you can see, for example, here, my students. Here, you see a string which is placed on the ground. Then, organisms touched by this line are recorded. You see they are recording the organism of their interest. That is line transect. My students, can you use a line transect in the field? It is simple to use. Let you have a loop, then place it on the ground. After there, organisms touched by that loop are recorded, are taken. Hope you are following, my students. Now, my students, we can have now the second type of transect, which is now called a belt. Belt transect. In a belt transect, there are two lines transect. Therefore, here there are two lines transect. Now, my student, these two lines run parallel. They run parallel. And they are separated from one another by a certain width. Let's say 0.5 meter or 1 meter apart. Are you following, my students? Now, for example, we can draw here. For example, this is one, this is another one. Therefore, this is the first line. Are we together? Then, this is the second line. You see, this line are running parallel to each other. Therefore, they are parallel running lines, two lines. There is a specific weight, for example, maybe of a half meter, 0.5 meter, my student. Then organisms in between these two lines are recorded. Therefore, organisms which are recorded are here in between, but at a regular interval. My student, you must have a specified distance. That is called belt transect. Now, my students, in a belt transect, the quadrant may be used, a quadrant frame. We will see later. For example, here, this is the first line. This is the second line. Therefore, the kind of this transect is called belt. A flame may be placed. For example, you place a quadrant flame here. Then organisms which are here are recorded. Then you place it here. Organisms which are here are recorded. Therefore, in belt transect, or line transect, quadrant flame is used. We'll see quadrant flame as we go on. My students, if you have got two lines now, you want to record the organism of a particular species, you can record, but which are in between the lines. Hope you are following, my students. Now, my students, can you use this in the field? Really, you can use. Let you have two strings, place them on the ground, select a width, maybe of 0.15, maybe of 0.5 or 1 meter apart. Then organisms in between the two lines are recorded. Hope you are following, my students. But sometimes you can record continuously, sometimes you can leave some area unsampled. Therefore, from there now, we can get two types of belt transect. Therefore, belt transect are of two types, my student. When an area is sampled continuously, we call continuous belt transect. Continuous what? Belt transect. Well, now, at a certain area, samples are taken continuously throughout the belt. For example, my student, you have a line here, you have a line here. Therefore, organisms here are taken continuously. They are recorded continuously. You can decide to put a flame quadrant here, for example, and here. You sample this area. Then you put it from here to here. You also sample this area. Then you put from here. Therefore, you continue continuously throughout. That is continuously belt transect. Therefore, no any space which is left unsampled. No any area or space is left unsampled. Therefore, the area is sampled continuously. Therefore, sample is taken continuously without leaving any part. Hope you are following, my student. But in some parts may be left unsampled. If a part is left unsampled, that is called interrupted belt transect. If we have also an interrupted belt what, my student? Transect. Therefore, here, my student, some parts remain unsampled. Some parts remains unsampled. Hope you are following, my student. Therefore, you can decide to leave a certain distance in between. Example, you have one line here, you have one line here. The first line, this is the second line. You can sample here. Let's say a sample is taken, my student, but this area remains unsampled. Then you sample here, sample is taken, the other area remains unsampled. Then you can sample here. Therefore, here you see some parts left unsampled. It depends on you. Which distance do you want to leave? You can leave a particular distance in between. Therefore, you can establish now a profile transect. Hope you are following, my student. Now, my student, I know you have a pen, you have a paper. Can you differentiate a continuous bed transect from interrupted bed transect? Question number 1. My students. Question number 2. Can you differentiate a line transect from a belt transect? Hope you are following, my students. Now, let us continue. My students, after knowing transect is now better, we know how to use quadrant. Quadrant is used. I said you can use transect and the quadrant both at the same time, but also quadrant may be used alone when you want to estimate a population size. Hope we are together, my students. Now, quadrants. What is a quadrant? For here, a quadrant, my student, is a frame of a known area. Therefore, a quadrant, these are frames of a known area. This frame may be made up of wooden material. Be made up of wooden materials, or may be made up of metal or PVC, polyvinyl chloride. A frame must have a known area. Therefore, a quadrant frame are of different sizes. We select a frame depending on the organism you want to sample. Hope you are following, my student. Therefore, a frame is placed on the ground. Therefore, this frame of a known area is placed on the ground, which can be thrown randomly on the ground. And this frame may be used along a transect line. Is used along a transect line. A frame, you find it used along a transect line, and organisms of interest are sampled. For example, you can see here my student. My student, you see this is a line, a transect line. Then a frame is placed here. Then species of interest are recorded. You can see they are recording species of interest. Are you following, my student? Therefore, a transect is there. Therefore, a transect line is there. A frame now of a known area can be placed along a transect line, and an ecological investigator conducting sampling. Is used along a transect line, and an ecological investigator conducting what is called sampling. Hope you are following, my student. My students, there are different types of quadrants. One of the type, my student, is called flame quadrant. This is a flame made up of wood, metal, or PVC materials, which is thrown randomly, and organisms within a flame are recorded. Therefore, this one here is made up of wooden materials or metal materials. This flame has a known area. Therefore, flame must have a known area. The selection of a flame depends on the kind of organism you want to sample. Therefore, selection of a flame depends on the organisms to be sampled. Hope you are following, my student. Now, flame quadrant may be square, rectangle, or sometimes cycle. But most flame quadrant, my student, are squares. Maybe rectangles, sometimes cycle. Therefore, you find a flame has several squares inside. Then these squares have a specified area. They have a specified area. You can, for example, see here, this quadrant flame has several squares inside. You can see the squares inside. These squares have a specific area. Hope you are following, my student. Okay? Now, okay, student, we can continue to see here. This is a flame quadrant. It's made up of metal or wooden materials. Then inside, there are squares made up of wire or maybe made up of string. These squares have a specific area. Together, my student? Now, depending on the size of organism we want to sample, you can select the suitable flame quadrant. For example, my student, you want to sample for lichens. Do you know lichen, my student? Association between fungi and algae. This one here is organism which is immobile. Immobile organism. If we want to sample here, select a quadrant with one side 0.5 meter and the other side is 0.5 meter. Therefore, the area here, the area of a square, you know, is side times what, my student? Side. Therefore, it will be now 0.5 meter times 0.5 meter. Here you get 0.25 meter square. Therefore, if you want to sample for lichen, select a quadrant with this area. If you want to sample for grasses, select a quadrant with 1 meter square. That means one side 1 meter, the other side 1 meter. If you want to get the area, take 1 meter times 1 meter, you get 1 meter square. My students, if we want to sample for shrubs, let's say, you know shrubs, the size of shrub is big than the size of grasses. Therefore, select a quadrant of 5 meter one side and 5 meter another side. Therefore, the area here will be 5 meter times 5 meter which you get now 25 meter square. Together, my student, if we want to sample area of woodland, maybe Miombo woodland, select a quadrant of 1 meter square or 400 meter square. Hope you are following my students. Therefore, quadrants are of different size depending on the organism which you want to sample. Now, my student, a flame quadrant is used when sampling immobile organisms. Examples of immobile organisms, my student, lichen, for example. Another, plants. And also when sampling small and slow moving animals. Small and slow moving animals. Therefore here, you can use when conducting random sampling. Therefore, it's used mostly in random sampling. How can you use this in random sampling? Take a flame, maybe of this kind, my student here, slow it randomly. Then organisms within a flame are recorded. Therefore here, a flame quadrant is thrown randomly, okay? Then organisms within a flame are recorded. Therefore, organisms within a flame are recorded. Hope you are following my students. Is that clear? Now, my student, you can use the flame quadrant together with the transect. As what I said before. For example, you have this is a point here. This is a point here. This is a point here. What is this type of transect, my student? I'm sure you know is Betty transect. Therefore, a flame may be placed here. This is a flame now. Now, organisms of your interest which are here are recorded. Then after there, you can place here a flame. And organisms of your interest present here are recorded. You continue throughout the area. Therefore, you must have a specified distance and also a specified area which you are sampling. But also, my student, it can be placed along a line transect. Now, if we go back to our picture here, this is a line transect. Now, a flame quadrant is placed. Then organisms of the interest are recorded. Hope you are following my student. Now, can you use flame quadrant when collecting the data of flora and fauna? It's simple, you can use. Take a flame, throw it randomly. Then organisms within a flame are recorded. Record them. Hope you are following my students. Now, my student, the second type of quadrant is called a pin flame. The second type is called a pin flame, or we call point quadrant, pin flame quadrant or point quadrant. Now here you have a crossbar Let's say this is our crossbar. This crossbar, you find a hose. Maybe this hose, maybe 10. Then this hose, a pin is placed. Therefore, you find a pin is placed. But this crossbar has legs which can be adjusted. Let's say these are legs which can be adjusted. Hope you are following my students. Then organisms here down which are touched by pin are recorded. Therefore, organism may be touched by this pin, and which are touched by pin are now recorded. Therefore, there a pin can hit the organism, but also a pin can miss the organism. Therefore, we have now there organism which can be touched, hits, and organism which can be missed by the pin. Therefore, a pin can miss the organism, a pin also can hit the organism. Hope you are following my student. But for this kind, you cannot use a pin quadrant when estimating the size, when conducting sampling of animals. We use a pin quadrant when you want to sample for grasses. You know, my student, grasses are difficult to be counted. Can you count grasses, my student? Really, you cannot count grasses. Therefore, here we can get the data by using a pin. Therefore, here what we will have is just the coverage of an area. Percentage cover of an area. Why do we use pin or flame quadrant? We use a pin or flame quadrant to organisms which are difficult to be counted. Hope you are following my student. You can see here, my student, this is a bar now having holes. You can see this hole, a hole here. There is also here. Therefore, in this hole is where a pin is placed. Therefore, this pin is placed here. Then the organism touched by the pin are recorded. Are we together? Hope we are together. But is only used for organisms which are difficult to be counted. Hope you are following my students. That is a pin flame or a pointy quadrant. Therefore, organism may be touched, may be not touched. Therefore, you will use it to calculate the percentage cover, of which now percentage cover will be equals to organism which touched by the pin of organism which touched by the pin plus which missed by the pin times 100. Are we together, students? For example, you have a bar, a flame having 10 holes. Have 10 holes. Then this one which is having 10 holes is used 10 times. Let's say is used 10 times. But the pins touch the organism only 36 units, maybe only 36. You are told calculate the percentage cover. Can you? Really, you can. Since a pin has 10 holes and is used 10 times, therefore it will be 10 times 10, you get 100, is it? Then the number of pins which touch the organism is only 36. Therefore, the percentage cover, which equals to hits, which is 36, over the total, which is 100 times 100. Therefore, here the percentage cover will be 36%. That is the percentage cover. Hope you are following my students. Then the last kind of quadrant is called permanent quadrant. Permanent quadrant, you find now here a flame is placed at a certain area permanently. Therefore here, flame is placed at a particular area but here permanently. Therefore here, you can keep on recording and recording. And then you can estimate the number of organisms based on seasonal variation. Because the flame is there permanently. My students, hope you can now explain properly how to use quadrant and also how to use transect. Is there any problem, my student? Uses of quadrant in estimating species distribution. My student, the uses now of quadrant and transect when you wanted to estimate species distribution. Uses of quadrants, let us start with. You can use quadrants when you want to know species density. My student, what is species density? Species density is the number of organisms of a particular species per unit area. Therefore here you take the total number of organisms of a particular species per unit area. Therefore here, if you are using quadrant, let's say we have a beta transect, we have another beta, we have a beta transect, my student. You put your flame quadrant here, then organisms which are here are counted of a species of your interest. Let's say you have counted maybe 20. I'm just letting these numbers, my student. Then, if you place a quadrant here, maybe you got only 5 organisms of your interest. You place a quadrant here, maybe you get 10 organisms of your interest. If you place a quadrant here, you get 15 organisms of your interest. Let's say from here to here. Together, student? You must have a specified distance, and here you must have a specified width. Okay? Now, student, let's say you are sampling an area where the length, I'm just letting my student, let's say the length is 100 meter and the width is 0.5 meter. 0.5 meter. Therefore, the area there will be 50 meter square. Together, my student. Then find the total number of organisms present. Therefore, calculate the total number of organisms. Let's say we have 20 according to what we have written here. 5, 10, 15. Now, student, you can have a calculator. Let us say we have here is it 20 plus 5 plus 10 plus 15. Eh? I'm getting 50. Therefore, student, number of organisms recorded equals to 50. But the area we know is 50 meter square. But you can convert this area into hectare. I'm sure you know, my student. You know 1 hectare equals to 10,000 meter square. How about 50 meter square equals to how many? Here you cross multiplication, you get 50 meter square times 1 hectare over 10,000 meter square. Therefore take 50 is 50 divided by 10,000. I'm getting 5 times 10 negative 3. Now is what? Hectare. This one is equal to 0.005 hectare. Now the species density which is equals to number of organisms over area. The number of organisms is 50 over an area which is 0.05 hectare. Therefore take that one, take 50 divided by your answer. Here I'm getting 10,000. Therefore you find is 10,000, which now organism per hectare. Together, my students? Okay, let us conclude by speaking about species frequency. My students, in species frequency we check on how times organism of a particular species occurred. Therefore we check on how many times organisms of a particular species observed. How many times? Therefore here we will check on the presence or absence of a particular organism or absence of a particular organism in an ecosystem. Hope you are following, my students. Take an example. We have now a transect. This is one line, this is another line. You know this type of transect is called Betty transect. And you have used a quadrant. Let's say in the first quadrant, organism of interest is present. In the second quadrant, organism of interest is not there, therefore is absent. In the third quadrant, organism of interest is present. And in the fourth quadrant, organism of interest is present. Can you calculate the species density? Really you can. Take the number of quadrants where organism of interest observed, divide by the total quadrant. Hope you can do this. Therefore, species frequency, my student, equals to number of quadrant where organism recorded. Therefore, you take a quadrant where organism of your interest recorded. Quadrants where organism of interest recorded. Recorded organism of your interest over total quadrants. Total quadrants, then times 100. Now from there, we can have now a species frequency, where in how many quadrants is species of interest recorded. From here, we get only 3 quadrants, 1, 2, 3. Therefore, only 3 quadrants where species of interest recorded. But the total number of quadrants studied were 4 times 100. Therefore here you get by 4, 1 by 4, 25. Therefore you get 75%. That is now the species frequency. Now student, that is the end of my presentation for today. I want to leave with you a question which you can do at home, explaining how ecological data are collected using quadrant. Hope you have enjoyed the lesson. Next lesson we will learn about the use of recapture in estimating population size. See you. Bye-bye.